The Science of Feeding Tomorrow. Pathway Intermediates.
So the scope of this webinar is quite broad. Hopefully will give you a background into it. I'm going to go over what is a microbiome and how microbiomes are comprised of commensal bacteria and how they interact with pathogens. Key to this is the actual function of the GI tract and how that is involved and interacts with the microbiome. And then I'll also cover why the microbiome is important, and I'll use examples in humans simply because we've studied ourselves the most. And then we'll also look at pigs and poultry, which is more specific to pathway work. And then I'll also cover how we at Pathway are looking at the microbiome and the technology that we're using as well. So what is the microbiome? This is a quote from the Microbiology Society blog, which defines a microbiome as a community of microorganisms living together in a particular habitat. And these microorganisms don't just live statically side by side, but they interact together, they communicate together and respond with each other. It's effectively an ecosystem. And the habitat in the human body can be the skin, the gastrointestinal tract, the gums, the teeth, and the feces. There are other examples as well. And each of these different habitats have different microbiomes. So the type of bacteria is different in each of these areas. Now, animals have co-evolved with microbial symbionts, so bacteria, and as a consequence, we have trillions of microbes that live inside our gut. Well, how do they get there? Initially, we thought that they were just transmitted vertically during the birthing process. However, more recent research has shown that they are transmitted from the mother via the placenta, and the main bulk of seeding does occur at birth. In poultry, it's slightly different because obviously these are chicks, they're hatched from an egg. And it's thought that the eggshell and the environment seed the gut microbiome. And there is some evidence, a recent evidence, that these are inherited, that some of the bacteria can be inherited as well. Now, the microbiome is influenced by many factors, and we'll go over some of those in a bit more detail today. But these include genetic, environmental, and key dietary factors. Diet has a huge influence on the gut microbiome. Now, the relationship that we have with our gut microbiome is typically beneficial to both the host and the microbe. And we use this term mutualistic symbiosis. It basically means that the two work together to benefit each other. However, this is a balance, and this can shift to pathogenesis. So the GI tract itself, we all know that it's involved in the digestion and absorption of nutrients from food and the excretion of waste. But it's also involved in immune surveillance. And with that, we have the gut-associated lymphoid tissue, or GALT, is key to that. And I'm going to go over these in a bit more detail shortly. But I wanted to start by looking at the GI tract itself, so we can have a look at the microbial abundances within it. So here we have a diagram of our gastrointestinal tract. Food starts off in the mouth, it gets chewed, it gets mechanically broken down to increase the surface area. The saliva in the mouth acts as a lubricant, and you also have enzymes, specifically amylase, which is present, and it starts the digestive process by digesting starch. This then moves through to the esophagus, where it enters into the stomach. Now, as you can see from this diagram here, we have a very small number of microbes in the stomach. This is because your stomach is very acidic. It's a hostile environment for bacteria to grow in. But this acidic environment is required for the liquefaction of the food, and it's here that you have a number of enzymes that are involved in protein digestion. Now, from the stomach, the food travels into the small intestine, but before and this is joined by secretions from two other organisms, two other organs. You have the liver, which produces bile salts, and this is involved in the emulsification of lipids in the food. And you also have the pancreas, which produces a number of enzymes such as lipases, proteases, and a pancreatic amylase, which has a similar function to the amylase in the saliva. These all enter the small intestine, which is where most of the nutrients are absorbed. And if you have a look at our diagram here, you can see that at the top of the small intestine, in the duodenum, we still have low levels of microbes, similar to the stomach. But as we travel down into the duodenum and the ileum, which are at the end of the small intestine, the number of microbes increases. And that's because the environment becomes less hostile and more hospitable to these microbes. The food then enters into the large intestine, or the colon, and this is where water absorption occurs, and is also the place where the most bacterial fermentation occurs, at least in humans. And as you can see here, we have the largest amount of bacteria. We have 10 to the 11 to 10 to the 12 colony-forming units per mil. So this is where bacterial fermentation takes place. So what does that actually mean? Why is that important? We have this long process of digesting and absorbing nutrients from food, but despite all of this, we can't digest it all. There are non-digestible fibres, such as carbohydrates, within the food that we cannot digest. And this is where our gut microbiome comes in. They have the enzymes, the proteins, that can degrade these into useful nutrients that we can then use. So an example of this is the non-digestible carbohydrates the microbes ferment these to produce short-chain fatty acids. And these are one of the main metabolites produced by the gut microbiome. Examples of these are butyrate, propionate, and acetate. And just to show how important these are, it's estimated that about 10% of calories obtained from the diet are from short-chain fatty acids, these metabolites produced by our gut microbiome. These would otherwise be excreted and wasted. And in fact, the colonic epithelium, so that's the cells that line the colon or the large intestine, derives up to 70% of its energy needs directly from butyrate, this short-chain fatty acid. So I think this shows quite nicely just how beneficial the gastrointestinal microbiota is for animals. Not only that, but the bacteria also ferment mucus that's present in the gastrointestinal tract already. So this is produced by the host, for example, us. We have our epithelial layer, which is lined by this mucus, and the mucus is a protective layer. And then above the mucus, you have the microbiome and the lumen of the colon. Now, certain bacteria can degrade the components within the mucus, and in fact, the composition of the microbiome is influenced by the glycosylation profile. So that's the type of sugars present on these mucus glycans. So the bacteria in the gut are also involved in protein metabolism, and they can synthesize amino acids. So amino acids we need to make proteins, but we can't produce them ourselves. We get them from the diet. And in fact, it's up to 20% of lysine and threonine, so these are two of the amino acids. 20% of these in non-ruminant animals are actually synthesized by gut microbes. So again, this just highlights how important these bacteria are to us. And they're also involved in the synthesis of vitamins, which are these essential micronutrients. And in fact, bacteria-derived vitamins, they can differ structurally from the dietary forms. I've got an example here of vitamin K. So vitamin K1 is found in green leafy vegetables, where vitamin K2 is produced by lactic acid bacteria. And these two vitamins we take in from the diet or the fermented products, but actually the vitamin K2, which is produced by the lactic acid bacteria, is easier for us to utilize and absorb and metabolize. And another example of the vitamins that bacteria can synthesize are the vitamin B family. So before, I also mentioned that the GI tract has an immune component to it. And this is very important if you think the immune system is involved in protecting us against microbes, and yet it has to work synergistically with microbes in our gut, commensal microbes that we want and in fact need to be there. And if we look at the gastrointestinal tract itself, it's generally considered to be external to the body, which takes a little while to get your head around. But if you think about it, you have basically a tube that goes from your mouth all the way through, and it has two openings that are open to the environment and so are available to pathogens. And it's one of the only regions in the body really where this occurs. So it makes sense that your gastrointestinal tract has lymphoid tissue or immune tissue available to protect the body and to protect the host. And in the GI tract, this is known as GALT, or gut-associated lymphoid tissue. And one example of the lymphoid tissue in the GI tract is called Peyer's patches. And these are discrete follicles found in the intestine that contain specialized cells. And these specialized cells are immune cells. They take up antigens and they deliver them to other immune cells to trigger an immune response. So the gastrointestinal tract has to be able to recognize antigens that are pathogenic and respond to them, but it also has to be able to tolerate antigens from food and from the microbiome. So there is this balance, this ability to be able to do this. It's quite unique. Now, the gut microbiota is also a first line of defense for us. It can protect us against pathogens itself by resisting the colonization of the gut by the pathogens. So how does it do that? There's only a finite amount of space for bacteria to be present, to colonize, and there's only a finite amount of nutrients for them to utilize. So if there are no spaces, because we have this commensal gut microbiota, then the pathogens can't colonize. There's no nutrients there, so they can't produce food, they can't feed, they can't grow. But also bacteria are in competition with each other. So different bacteria produce antimicrobial factors against other bacterial species. So you have this almost fight between the bacteria themselves to colonize the GI tract. And an example of these antimicrobial factors are bacteriocins. And these are a protein released by one type of bacteria that target another, and they produce holes inside the cell membrane. So they cause the cell to lyse and it destroys the cell. And the gut microbiota is also hugely important in the development of the immune system. And there have been a number of studies that are required for that have shown how important this is and how important the gut microbiome is in developing this tolerance that I've touched on briefly. So it can go wrong, of course it does. That's when we get diseases. Aberrant interactions are linked to of the gut microbiome and the host are linked to inflammatory bowel disease. Stress can also change the organisms in the microbiota, and in fact the microbiota has been shown to affect the stress response. And changes in the diet can lead to changes in the gut microbiome. For example, diets in humans, diets high in saturated fats lead to an increase in this particular bacteria, which has been shown to increase susceptibility to colitis or inflammation. And antibiotics, the antibiotics that we use tend to be broad spectrum, but they're not able to discriminate between the good, beneficial, commensal bacteria that we use and pathogens. So this can have a huge effect on the microbiome itself. So I thought I'd move on now to look at pigs and the GI tract in pigs. And I showed you in the humans that we have different microbial abundances in different areas of the tract and of the GI tract. And that's true in the pigs as well. But what's also true is that we have a different composition of microbes in the different organs within the GI tract. So what we have here is a diagram of our pig. And this shows the different phyla of bacteria. And what you can see is that the main phyla is Firmicutes. And this phyla contains bacteria that are involved in energy reabsorption, so production of these short chain fatty acids that we spoke about earlier. And what you can see is if, for example, you take the stomach, yes, we have a large amount of Firmicutes. We also have proteobacteria here as well. So then if we move over to the cecum, still dominated by Firmicutes, but then we have this switch here, where it's now the bacteridietes rather than the proteobacteria that are present. So we have those are examples of spatial changes in the gut microbiome. So that's different microbiome composition depending on location. We also have temporal changes, which means that the gut microbiome changes and develops as the animal ages. And there are many factors, as I've said before, that affect the GI microbiome. And in swine, probably one of the most important ones is stress, particularly that associated with weaning. Now, weaning of the piglets is a huge challenge for the swine producers. You have this dietary shift from a high-fat, low-carbohydrate diet to pretty much the opposite, a low-fat, high-carbohydrate diet. And this triggers a microbial shift. So you move from milk-fermenting bacteria to plant fermenting bacteria. And in addition, you expose the piglets to new bacterial species, including pathogens. And what can happen within weaning is you get post-weaning diarrhea, which occurs within 2 weeks post-weaning, and you see dehydration, diarrhea, significant mortality, and loss of body weight. So let's have a look at how this affects the GI tract. The GI tract microbiome. So as I've said, it's this change in diet, and because you're changing the piglet's diet, they stop to eat. They stop eating as well. So you get this drop in feed intake, and you expose them to new bacterial species. But this is important for the growth and development of the piglet immune system, so it is an important step. But this stress that's associated with it, and the anorexia that occurs, so when they stop to eat food, you see actually triggers a change in the morphology of the intestine. And what this means is this change in morphology actually compromises the mucosal barrier. So that's the barrier where you have the epithelial cells and the mucus. And this change, compromising this barrier allows antigens to enter, and this triggers an inflammatory response. Excuse me 1 second, I have to cough. Thank you, sorry about that. So this inflammation also triggers morphological changes, and these affect the nutrient digestion and absorption, and affect the function of the intestinal barrier. And one of the things that this triggers is the innate immune system. And the immune system's response is to secrete something called free radicals. This is part of the body's immune response mechanism. It's a perfectly normal and natural response to it. However, sometimes it doesn't go according to plan. And you have these free radicals that react together in the gut lumen, producing nitrate. Now, nitrate in the gut is then available for bacteria to utilize, and bacteria will use whatever is available for them. And in a nitrate-rich environment, bacteria such as S-type Salmonella typhimurium and enterotoxigenic E. coli will use these. And these pathogens induce further intestinal inflammation, disrupts the microbiota further, it disrupts the intestinal morphology further, and you get this cycle of inflammation. And so this is one of the causes of post-weaning diarrhea, but it's not the only potential one. This is just an example involving the gut immune system and the gut microbiota. So if you have a look at the poultry GI tracts, again, we have different abundances of bacteria within the GI tract. What's interesting about the poultry is that in, for example, humans, we have the main bulk of our fermented bacteria in the colon. In poultry, it's in the ceca. So the ceca are these two organs here, and there they have 10 to the 10 or 10 to the 11 cells per gram. So this is where the main bulk of their bacteria is. But again, you can see the different types of bacteria that are present in the different organs. So that's that spatial distribution, the changes in the microbial composition throughout the GI tract. Now, I quite like this image here, taken from the paper, because it shows just how many different factors influence the microbiome. The specific example here is in poultry, but it holds true for many animals as well. So we have genetic factors that can affect these. You've got the bird species, the type, whether it's a broiler or a layer, the breed, even the sex of the chicken affects the host microbiota. And environmental factors, seasonal changes, and feed, litter management, nutrition, and supplements that we add into and out of the diet all affect the gut microbiota. And you want to get that balance right between a healthy chicken and productive performance, where you see a good growth rate. And I think this highlights that quite nicely. But I've said before about temporal changes within the chickens, within the GI tract. And this is, again, it's an image taken from a paper. And we have this is the cecal microbiome. So the microbiome found within the chicken ceca, which, as I said, is the organ with the most abundant microbial population. And you can see from week 1 and week 12, that there is an increase in the types of bacteria. So what we have here is, this is one sample in week 12. The blue bar is one type of bacteria. Then we've got the green, which is another, and these, all the different colours represent different bacterial species. So this blue species does dominate, but we see an increase in the number of other bacteria as the chicken ages. And this is just as the chicken matures, as the diet changes, it becomes more diverse. So we see a greater number of bacterial species present, and it tends to become more stable as well. So how exactly do we look at this? How do we at Pathway look at this? So this is just an overview slide. We take our trial, our poultry trial, we collect samples from the chickens, we prepare them, we sequence them, and then we analyse them. So with the trial, we work with the colleagues in South Korea, where we have the Biomedical Research Institute. They have a research farm, and we they design poultry trials, typically 5 to 6 weeks, where we look at different dietary groups. We compare them to a control group. We collect the samples for sequencing, and as I've said before, different intestinal sites have different microbial abundances. So you have to pick your sampling site very carefully, and it very much depends on the question that you're trying to ask. So you can take fecal samples. The pros of doing this is it's easy, it's convenient, it's non-invasive, and it's relatively inexpensive. However, the cons of this is that you can get bacterial contamination from, for example, the litter. There might be uneven bacterial distribution, and it's not always representative of the gastrointestinal tract. Cecal samples are quite commonly used, and they're particularly useful in birds because, as we've seen, the ceca has the densest population of microbes. And when sampling those, you can control the contamination because you're not collecting it from the litter. Cons, it's typically invasive and you get small samples in young birds, and it is a typically endpoint sample. You can look at other intestinal sites. Pros, you can do the spatial analysis, and there is also this contamination control, but again, it's invasive, it's endpoint, and it can be expensive. And as I've said, the sampling site that you pick and that you use, it very much depends on the question of the trial that you're holding. So then we have to prepare the sample. So good quality data needs good quality samples. And what we do is we actually extract the DNA within that sample. We have to purify it. And then we selectively target the bacterial DNA. So that's quite important because when you get your DNA sample, you've taken it from another animal, from a chicken. So you're going to have a lot of chicken DNA in there as well. By selectively targeting the bacterial DNA, we know that we're looking at the microbiome specifically and not contaminating DNA. So the way we do this is a technique called a polymerase chain reaction, where we take the DNA and we amplify up a specific gene that is specific to bacteria. And this is what this diagram here is showing. So we have this specific bacterial gene. We add what are called primers to it. These bind to it, and we can then amplify up this region, this portion here, and we get lots of copies of that gene. What this means is we can take a very small DNA sample and we can amplify it to get a large amount of DNA, and it maintains patterns because it's so specific and it's very sensitive. It maintains patterns in the abundance levels of the bacteria from the sample. Another thing we do is these primers that we use to amplify up the gene, they contain these unique identifiers, and this allows us to pool our samples. It makes it cost and time efficient, and it also means that our samples are maintained and we can treat them in exactly the same way as we move on to the sequencing side of things. So then we sequence our DNA sequences, and we get these reads of DNA, and then we use a computer program to assign the taxonomy to them. So we put these DNA sample sequences through, and we identify which bacteria are present in the sample. And this here is an example of that. So we get our bacterial taxon, we get the number of reads through, and we can also get a taxonomic tree that comes out of this and tell us what bacteria are present. This data is data taken from a control. So this is a commercially available community control that we use to check our sequencing data to make sure it's consistent and reliable and matches up with what we want it to. And then we analyse this data. There's a lot of quality control that goes into it. We have our internal controls itself. And from that data, we can calculate the relative abundances of bacteria within a sample. So we can look at the proportion of which bacterial genuses are present in our particular sample. And again, this is example data from our mock community control. So what we have here is we have the relative abundance of different bacterial phyla. We have firmicutes in the blue and proteobacteria in the yellow. And we have 4 separate runs that we did to check to see if we were getting consistent results, which we were. And then we looked at this at the genus level as well. So same data set, but just looking at the different bacterial genera. And this is an example of temporal changes in the broiler microbiome. So again, this is a data set from work that we've done. We looked at the cecal microbiome at day 7, day 21, and day 35. And as you can see, yes, we have a number of bacterial species at day 7, but this increases as we move on to day 35. And probably this section here constitutes other bacteria. And this is our cut-off point where we pool bacteria together with an abundance of less than, I think it's 1%. And we have, this shows that we have an increase in diversity. So we're getting more bacterial species as the chickens age, which is what's been seen before. And then this is just an example from a trial that we took. We had 3 trial diets, a control diet and 2 test diets. And we looked at the microbiome at week 1 and week 6. And whilst our trial diet number 1 looked very similar to the control, we do have some changes, but it's very, very similar. Trial diet 2, we can detect quite significant changes in that. And hopefully this just gives you an idea of the kind of capabilities that we have here at Pathway in sequencing the gut microbiome.